A human body is not a single unified structure in the way we often imagine it. It is a layered system of organization, built from levels upon levels of smaller components. Each level follows rules that are simpler than the level above it, yet together they create something far more complex. When we begin to study life closely, we do not start with the whole organism. We start with smaller and smaller scales. Cells are one of these fundamental scales. And inside cells, there are structures that carry out specialized roles continuously. One of these structures is the mitochondrion. It is not large enough to see without magnification, yet it is present in nearly every cell of the human body. Its presence is not occasional, it is constant. And its activity is not optional. It is required for life as we currently understand it. Before focusing on mitochondria directly, it is helpful to slow down and consider scale itself. Scale is not just a measurement, it is a way of organizing understanding. At the largest scale, we observe the human body moving through space, interacting with the environment. At a smaller scale, we observe organs coordinating function. At a smaller scale again, we observe tissues composed of repeating units. And at the cellular level, we begin to see independent living units performing internal processes. Each step downward in scale reveals not simplicity, but different kinds of complexity. Complexity does not disappear when we zoom in. It changes form. A single cell is often described as the basic unit of life. This is not because it is simple, but because it is self-contained in many of its functions. A cell maintains boundaries. It regulates internal conditions. It processes nutrients. It responds to signals. But even this description is incomplete without acknowledging internal specialization. Inside a cell, different structures perform different roles. Some handle information, some handle transport, some handle structural support, and some handle energy transformation. Energy is a central requirement for all living systems. Without energy, processes stop. Without controlled energy, organization breaks down. Cells do not use energy in a vague sense. They rely on a specific molecule called ATP, adenosine triphosphate. ATP is not stored in large reserves for long periods. It is continuously produced and continuously consumed. This creates a cycle of production and usage that must remain stable. Mitochondria are central to this cycle. They are structures within cells that convert chemical energy from nutrients into usable ATP. This process is not instantaneous. It is step-based. It involves a sequence of chemical reactions that gradually transfer energy from one form to another. Each step is controlled. Each step is regulated. To understand mitochondria clearly, it helps to avoid imagining them as simple power generators in the mechanical sense. They are not like electrical outlets or batteries in a direct analogy. Instead, they are dynamic biochemical systems. They participate in multiple pathways simultaneously. They respond to the needs of the cell. They adjust output based on demand. The term powerhouse of the cell is often used as a simplified description. This phrase is useful as a starting point, but it does not fully describe their behavior. Mitochondria are better understood as energy processing centers embedded within a larger system of cellular regulation. They do not act alone. They interact with enzymes, membranes, and signaling molecules. Their function depends on context. If we zoom into a single cell and slow observation further, mitochondria appear as small, oval, or elongated structures. They are surrounded by two membranes. This double membrane structure is significant. It is not arbitrary. The outer membrane separates the mitochondrion from the surrounding cytoplasm. The inner membrane contains folds that increase surface area. These folds are essential for energy-related reactions. However, at this stage, we are not yet focusing on structure in detail. We are focusing on the idea of presence. Mitochondria are present in varying numbers depending on the cell type. Cells that require more energy, such as muscle cells, contain many mitochondria. Cells with lower energy demands contain fewer. This distribution is not random. It reflects functional needs. The presence of mitochondria in nearly all human cells suggests a shared dependency. Regardless of cell type, energy is required for survival and function. This shared requirement creates a unifying pattern across biology. Different systems, same dependency. At this point, it is useful to return to the idea of scale once more. From the outside, a human appears continuous. Movement appears smooth. Thought appears immediate. But inside, processes are discrete and structured. Energy must be extracted, converted, transported, and used. Mitochondria are part of this internal structure of conversion. They are not isolated units acting independently in a chaotic environment. They are embedded in a regulated system. They communicate with the rest of the cell through chemical signals. They respond to nutrient availability. They respond to oxygen levels. They respond to cellular demand. This responsiveness is important. It means that mitochondria are not simply on or off. Their activity exists along a spectrum. At times of high demand, they increase output. At times of low demand, they reduce activity. This regulation helps maintain stability inside the cell. Stability is a key concept in biology. Living systems are not static. They are dynamic. But this dynamic behavior must remain within limits. If energy production is too low, function fails. If energy production is too high without regulation, imbalance occurs. Mitochondria participate in maintaining this balance. At a chemical level, mitochondria are involved in converting nutrients into ATP through a series of reactions. These reactions involve electrons, protons, and molecular carriers. The details of these processes will be explored later. For now, it is enough to understand that energy is not created directly. It is transferred and transformed. This transformation process is gradual. It is structured into stages. Each stage depends on the previous one. This creates a chain of dependency. If one step is disrupted, the overall process is affected. Because of this dependency, mitochondria must maintain internal organization. Their membranes, enzymes, and internal compartments are arranged to support sequential reactions. This organization is not static. It is maintained through continuous regulation. At this point, we are still observing mitochondria from a distance. We have not yet entered their internal structure in detail. We have not yet followed the chemical pathways step by step. Instead, we are building a conceptual foundation. This foundation is important because biological systems can appear overwhelming when viewed only through detail. By first understanding scale, presence, and function, we create a framework for deeper exploration. Mitochondria are often introduced early in biology education, but their significance extends beyond a simple label. They represent a connection between chemistry and life. They represent the interface where molecular energy becomes biological activity. This interface is continuous. It does not pause when we are resting. It does not pause during sleep. Even when the body is still, cellular processes continue. Energy production continues at a reduced rate, adjusted to current demand. In this way, mitochondria are always active in the background of life. Not as isolated engines, but as part of a distributed system of energy regulation across trillions of cells. As we prepare to move deeper into cellular structure and organization, it becomes useful to reframe the cell itself as a coordinated environment rather than a single entity. Within this environment, mitochondria occupy a specialized role that connects chemistry, structure, and energy flow. The next step is to slow down further and look at the cell not as a biological unit, but as a quiet, structured environment, almost like a small city with repeating patterns of organization. A cell can be understood as a small, self-contained system of organization. But it is more accurate to describe it as a structured environment where many processes occur at the same time. These processes are not random, they are coordinated. Each component inside the cell has a role that contributes to overall stability. To make sense of this, it helps to use a simple analogy, not as a literal comparison, but as a way to organize thought. A cell can be imagined as a quiet city. Not a city of noise or movement in the human sense, but a city operating at a microscopic scale, where everything is slow, continuous, and regulated. In this city, different regions perform different functions. There are areas responsible for information storage, areas responsible for energy processing, areas responsible for transport, and areas responsible for structure. But unlike a human city, these regions are not physically separated by distance. They exist within the same enclosed boundary. The boundary of the cell is the cell membrane. This membrane is not a rigid wall. It is a selective interface. It allows certain substances to pass while restricting others. It maintains balance between the internal environment and the external environment. This balance is essential. Without it, internal processes would lose stability. The membrane is not passive, it is active in regulation. It responds to signals and controls exchange. Inside this boundary, the internal space of the cell is called the cytoplasm. The cytoplasm is not empty. It is filled with fluid, molecules, and structures suspended in a controlled environment. Within this space, biochemical reactions occur continuously. If we imagine the cell as a city, the cytoplasm is the open working space. It is where transport occurs, where molecules move, where reactions take place. But even this movement is not chaotic. It is guided by gradients, signals, and structural pathways. Within this environment are organelles. Organelles are specialized structures that perform specific tasks. Each organelle is enclosed or organized in a way that allows it to maintain its function. The nucleus is one of the most prominent organelles. It contains genetic information in the form of DNA. This information is not constantly active in full. Instead, it is accessed in regulated segments depending on cellular needs. The nucleus can be thought of as a control center, but this is also a simplification. It does not directly perform all actions. Instead, it provides instructions that are interpreted by other parts of the cell. These instructions are transcribed into RNA and then translated into proteins. Proteins are functional molecules that carry out most cellular tasks. They form structures, catalyze reactions, and regulate processes. But even this system of information and execution depends on energy. Without energy, no process can proceed. This is where mitochondria becomes central again, even within the broader city analogy. Mitochondria are distributed throughout the cytoplasm. They are not located in a single central area. Instead, they exist in many locations, positioned where energy demand is high. This distribution is functional. It ensures that energy can be supplied locally rather than transported over long distances inside the cell. In muscle cells, for example, mitochondria are densely packed because contraction requires continuous energy. In neurons, mitochondria are also abundant, especially in regions where signals are transmitted. This variation shows that the cell is not uniform. It is adapted to function. Returning to the city analogy, mitochondria can be thought of as distributed energy stations. Not a single central power plant, but many small systems placed strategically throughout the environment. However, unlike machines in a city, mitochondria are not independent from the rest of the system. They respond to signals from the nucleus and from the cytoplasm. They adjust their activity based on demand. This responsiveness is critical. It means that energy production is not fixed. It is adaptive. To understand how this adaptation fits into the cell, we can observe the flow of materials. Nutrients enter the cell through the membrane. These nutrients are broken down into smaller molecules. These molecules are then transported to mitochondria for further processing. This flow is continuous. It is not a single event. It is a steady exchange of matter and energy. Inside the cytoplasm, transport occurs through diffusion and guided movement along structures called the cytoskeleton. The cytoskeleton is a network of protein fibers that provide structure and pathways for movement. This network allows organelles to remain organized within the cell. It also allows for movement of vesicles and molecules between different regions. In this way, the cell is not static. It is constantly reorganizing at a microscopic level. Even though the cell appears stable from a macroscopic view, its internal environment is dynamic. Molecules are constantly being synthesized, broken down, transported, and recycled. Mitochondria exist within this dynamic environment and must adapt continuously. They are not isolated from cellular motion. They are part of it. One important feature of cellular organization is compartmentalization. Even though everything exists within a single membrane, internal separation still occurs through organelles. This separation allows different chemical environments to coexist without interference. Each organelle maintains conditions suitable for its function. For mitochondria, this includes maintaining gradients across membranes and controlling internal chemical conditions. These gradients are essential for energy production, which will be explored later in detail. For now, it is enough to understand that mitochondria rely on controlled differences in concentration across space. Within the cell city analogy, this can be thought of as controlled differences in pressure or potential between regions. These differences are not accidental. They are maintained actively. The maintenance of these differences requires energy itself. This creates a recursive relationship. Energy is required to maintain the system that produces energy. This is one of the fundamental characteristics of living systems. They are self-sustaining through continuous regulation. At the level of the cell, regulation is achieved through feedback loops. If energy levels are low, signals increase mitochondrial activity. If energy levels are high, activity is reduced. This prevents instability. It ensures balance between supply and demand. In addition to energy regulation, mitochondria also interact with other processes in the cell. They are involved in calcium signaling, metabolic signaling, and even pathways related to cellular lifespan. This means that mitochondria are not only energy providers, they are also integrators of cellular state. The idea of integration is important. It suggests that mitochondria contribute to decision-making processes at the cellular level, not in a conscious sense, but in a biochemical regulatory sense. They respond to stress signals. They respond to nutrient availability. They respond to damage signals. In some cases, mitochondria are involved in initiating programmed cell death, a controlled process that removes damaged or unnecessary cells. This function highlights their role beyond energy production. Within the cell city analogy, this is similar to regulated shutdown or restructuring of certain systems when conditions are not sustainable. However, it is important to maintain clarity. These analogies are tools for understanding, not literal equivalents. Cells do not operate with intent or awareness. They operate through chemical and physical interactions governed by molecular rules. Still, the analogy helps organize the complexity into understandable layers. As we observe the cell more closely, we begin to see that its stability depends on constant activity. Nothing inside the cell is truly idle. Even structural components are maintained dynamically. Mitochondria within this system remain active participants in sustaining life at the cellular level. They are not separate from the rest of the cell's processes. they are embedded within them. As we move forward, it becomes necessary to zoom in further. The next step is to leave the broader organizational view of the cell and begin examining how mitochondria are structured internally, including their membranes and compartments. The next section moves inward into the layered structure of the mitochondrion itself, where energy transformation begins to take a more defined form. A long time before complex life forms existed, before plants, animals, and human beings, the Earth was populated only by simple, single-celled organisms. These early life forms were structurally simple compared to modern cells, but they were already capable of sustaining themselves, reproducing, and adapting to their environment. At that stage in Earth's history, life was entirely microscopic. There were no multicellular organisms, no nervous systems, no organs, only individual cells interacting with their environment. Yet even in this early world, energy conversion was essential. Every living system requires energy to maintain internal order. Without a mechanism to process energy, even the simplest cell cannot sustain itself. At some point in evolutionary history, a major transition occurred. One cell entered another cell, and instead of being destroyed, it remained. Instead of competition, there was persistence. Instead of digestion, there was cooperation. This event is known as endosymbiosis. It describes a relationship where one organism lives inside another in a mutually beneficial arrangement. In this case, the internalized organism was an early form of bacteria capable of efficient energy conversion. The host cell was a larger, single-celled organism that could provide protection and access to nutrients. Over time, this relationship became stable. The internal organism was no longer independent in the way it once was. It became integrated into the host cell's internal structure. This integration did not happen instantly. It occurred gradually over evolutionary timescales spanning millions of years. As the relationship stabilized, the internal organism began to lose some of its independence. It transferred many of its functions to the host cell while retaining core energy-producing capabilities. The host cell, in return, provided a stable environment and a continuous supply of nutrients. This mutual dependence became so strong that neither system could easily exist without the other. Over time, the internal organism evolved into what we now call the mitochondrion. This origin story is not mythology. It is supported by multiple lines of biological evidence, including the presence of mitochondrial DNA, double membranes, and similarities to bacterial structures. However, for the purpose of understanding mitochondria, what matters most is not the historical narrative itself, but what it reveals about function. Mitochondria are not simply parts of a cell. They are the result of integration between two once separate biological systems. This means they retain characteristics that distinguish them from other organelles. One of these characteristics is the presence of their own genetic material. Unlike most organelles, mitochondria contain DNA. This DNA is separate from the DNA found in the nucleus. However, mitochondrial DNA is limited. It does not encode all the proteins mitochondria require. Many mitochondrial proteins are encoded by nuclear DNA and transported into mitochondria after being produced in the cytoplasm. This creates a coordinated relationship between nucleus and mitochondria. One provides instructions. The other performs specialized energy-related functions. This coordination is essential for cellular function. It reflects a division of labor within the cell. Returning to the origin of mitochondria, the idea of symbiosis is important because it reframes how we think about cellular components. Instead of viewing organelles as purely constructed elements, we can see them as integrated systems with historical continuity. Mitochondria are not artificial structures built from scratch. They are the result of evolutionary integration. This integration had profound consequences for life on Earth. Before mitochondria became part of complex cells, energy production was relatively limited in efficiency. After their integration, cells gained access to significantly more efficient energy conversion processes. This increase in energy availability is believed to have enabled the evolution of more complex organisms. Larger cells, multicellular structures, and eventually specialized tissues all depend on sufficient energy supply. In this sense, mitochondria are not just components of life. They are enabling structures for biological complexity. To understand why this is significant, it helps to consider energy as a limiting factor in biological organization. Systems can only become more complex if they can support the energy demands of that complexity. Mitochondria increased the energy capacity of cells, which allowed new levels of organization to emerge. But the origin story also introduces another important concept: internal boundaries. The mitochondrion is enclosed by two membranes. This double membrane structure is a direct reflection of its evolutionary origin. The outer membrane corresponds to the original engulfing boundary of the host cell. The inner membrane corresponds to the original membrane of the engulfed organism. Between these membranes is a compartment called the intermembrane space. Inside the inner membrane is the mitochondrial matrix. These compartments are not arbitrary. They are essential for energy conversion processes that occur later in detail. For now, it is enough to understand that mitochondria are structured with internal separation that reflects both function and origin. This structure allows mitochondria to maintain chemical gradients, regulate internal conditions, and carry out sequential reactions. Gradients are particularly important. They represent differences in concentration between compartments. These differences store potential energy that can later be converted into usable biological energy. Without compartments, gradients would not be stable. Without stable gradients, energy conversion would not be efficient. Thus, structure and function are tightly linked. The mitochondrial membranes are also selectively permeable. This means they control what enters and exits. Not all molecules can pass freely. Specific transport mechanisms are required. This selective transport allows mitochondria to maintain internal conditions distinct from the surrounding cytoplasm. Inside the mitochondrial matrix, many enzymes are present. These enzymes facilitate chemical reactions involved in energy production. Enzymes are not consumed in these reactions. They function as catalysts, enabling reactions to occur efficiently without being permanently altered. This catalytic environment is essential for sustaining continuous energy conversion. The presence of mitochondrial DNA adds another layer of autonomy. Although mitochondria depend on the cell for many proteins, they retain partial genetic independence. This partial independence reflects their evolutionary past. However, over time, most of their original genetic material has been transferred to the nucleus. This process is known as gene transfer. As a result, mitochondria are now highly dependent on nuclear genes for their function. This creates a distributed genetic system within the cell. No single compartment contains all information required for mitochondrial function. Instead, information is shared across cellular structures. This interdependence is a defining feature of eukaryotic cells. It also highlights a broader principle in biology. Systems evolve toward integration when cooperation provides a survival advantage. The endosymbiotic origin of mitochondria is one of the clearest examples of this principle. What began as a separate organism became embedded within another system. Over time, boundaries blurred. Functions became shared. Identity became integrated. Yet traces of the original separation remain visible in structure and genetics. This dual nature is important for understanding mitochondria today. They are both independent in certain molecular features and dependent in overall function. As we observe mitochondria within modern cells, we are observing the outcome of a long evolutionary process of integration and optimization. Their structure reflects history. Their function reflects necessity. At this stage of understanding, we can see mitochondria not only as energy processing units, but as evolutionary artifacts embedded within every cell. They carry within them the memory of ancient biological cooperation expressed in modern biochemical form. As we move forward, we will begin to look more closely at how this structure supports energy conversion at the molecular level. We will examine how nutrients are processed, how electrons are transferred, and how gradients are formed and used. The next section moves from origin and structure into the internal architecture of the mitochondrion itself, where energy transformation begins to take a more defined and mechanical-like biochemical form. Inside the mitochondrion, structure becomes more specialized and more functionally organized. What appeared earlier as a simple oval or elongated shape now reveals itself as a layered internal system. This internal organization is not decorative. It is essential for how energy is processed. The mitochondrion is enclosed by two membranes. These membranes are not identical. They differ in composition, permeability, and function. The outer membrane forms the boundary between the mitochondrion and the cytoplasm. It is relatively permeable compared to the inner membrane. This allows certain molecules to pass more easily into the space between the membranes. The inner membrane is far more selective. It tightly regulates movement of substances. It is highly folded, forming structures known as cristae. These folds increase surface area significantly. This increase in surface area is not incidental. It directly supports energy conversion processes that occur on the membrane. Between the outer and inner membranes lies a narrow space called the intermembrane space. This region is important for maintaining chemical differences between compartments. Inside the inner membrane lies the mitochondrial matrix. The matrix is a dense, enzyme-rich environment where many metabolic reactions occur. These three regions together form a structured internal system. Each region contributes to energy production in a different way. To understand mitochondria clearly, it is important to view them not as single compartments, but as coordinated, layered systems. Each layer supports a different aspect of energy transformation. The inner membrane is the most functionally significant region for energy conversion. It contains protein complexes embedded within its structure. These complexes are responsible for transferring electrons and moving protons across the membrane. This movement of particles is not random. It is organized into sequential steps. Each step contributes to the gradual buildup of a gradient. Before energy can be stored in a usable form, nutrients must be broken down. This breakdown occurs through earlier metabolic pathways, but mitochondria complete key stages of the process. Molecules derived from glucose and other nutrients enter mitochondria in processed forms. These molecules are further oxidized, meaning they lose electrons in controlled reactions. Electrons are not lost into empty space. They are transferred between molecules and protein complexes in a structured pathway. This pathway is located in the inner mitochondrial membrane. It is known as the electron transport chain. Before examining this chain in detail, it is important to understand the physical environment in which it operates. The inner membrane is not a smooth surface. It is folded into cristae, which extend inward into the matrix. These folds create additional surface area for embedding protein complexes. More surface area means more sites for energy-related reactions. This is a structural optimization. It increases efficiency without increasing volume. Within the membrane, several large protein complexes are arranged in sequence. These complexes are labeled as complex 1, 2, 3, and IV in simplified descriptions. Each complex performs a specific step in electron transfer. Electrons move from one complex to another through intermediate carriers. As electrons move through this system, energy is gradually released. This energy is not released all at once. It is captured in small increments. One of the key outcomes of this energy release is the movement of protons across the inner membrane. Protons are positively charged particles. Their movement creates a difference in charge and concentration between compartments. Specifically, protons are moved from the matrix into the intermembrane space. This creates a higher concentration of protons outside the matrix relative to inside. This difference is not static. It is actively maintained by continued electron transfer. The result of this process is the creation of an electrochemical gradient. This gradient represents stored potential energy. It is important to emphasize that mitochondria do not store energy directly as ATP at this stage. Instead, they store energy in the form of spatial and chemical differences. These differences are then used to drive the final step of ATP production. The inner membrane, therefore, functions as a barrier and a conversion surface at the same time. It separates compartments while also enabling controlled transfer of energy. The intermembrane space plays a supporting role in this system. It acts as a reservoir for protons. It temporarily holds charge differences created by electron transport. The matrix, on the other hand, is the environment where many metabolic reactions occur. It contains enzymes responsible for processing molecules into forms usable by the electron transport system. These three regions, outer membrane, intermembrane space, and matrix, work together in a coordinated system. Each region maintains specific chemical conditions. Each region contributes to overall energy transformation. The double membrane structure is therefore not just a boundary. It is a functional framework for energy conversion. If we slow down further and observe the inner membrane conceptually, we can see it as a surface where motion becomes structure. Electrons move along protein complexes, and this movement is coupled to proton transport. This coupling is essential. Without it, energy would not be efficiently captured. Instead of being released as heat or lost, energy is stored in a controlled gradient. This gradient is maintained as long as electron transfer continues. When electron flow slows or stops, the gradient dissipates. This highlights the dynamic nature of mitochondrial energy systems. Nothing is permanently stored in isolation. Everything depends on continuous activity. Within the matrix, additional metabolic cycles take place. These cycles generate electron carriers that feed into the electron transport chain. These carriers act as intermediates. They transport electrons from earlier stages of metabolism into the membrane-bound complexes. This creates a continuous flow of energy-related particles through the mitochondrion. The coordination between matrix reactions and membrane processes is tightly regulated. If one part slows, the other adjusts accordingly. This coordination ensures stability in energy production. It is also important to note that mitochondria are not isolated from the rest of the cell. They exchange signals with the cytoplasm and nucleus. These signals regulate how much energy is produced. They respond to cellular demand in real time. If the cell is active, mitochondria increase activity. If the cell is at rest, mitochondrial activity decreases. This regulation prevents waste and maintains balance. The structural organization of mitochondria makes this regulation possible. Without membranes and compartments, gradients could not be formed or controlled. Thus, structure and function are inseparable. At this stage of understanding, we can see mitochondria as layered systems of controlled separation and controlled connection. They separate environments to create gradients. They connect processes to transfer energy. This duality is central to their role in life. As we continue deeper, the next step is to focus on the chemical cycle inside the matrix that begins the process of electron generation. This cycle is circular, continuous, and foundational to mitochondrial energy flow. The next section moves into the matrix-based metabolic cycle that sustains electron production and prepares energy for conversion through the inner membrane system. Inside the mitochondrion, energy production does not begin at the membrane. It begins deeper, within the matrix, where small molecules are continuously processed through a repeating cycle of reactions. This cycle is not linear. It does not move from a single start to a single finish in a straight path. Instead, it forms a loop, a continuous return to starting conditions, ready to begin again. This cycle is known in biology as the Krebs cycle or citric acid cycle. But here, it is more useful to think of it as a structured transformation loop, a sequence where molecules are gradually reshaped and energy is carefully extracted in small steps. At this level, nothing happens suddenly. Every change is incremental. Every transformation is controlled. To understand this cycle, we begin with a simple idea. The cell receives nutrients from its environment. These nutrients are broken down into smaller molecules before they reach the mitochondrion. One of the key molecules entering the cycle is acetyl-CoA, a small carbon-based compound. Acetyl-CoA does not remain unchanged. It enters a structured sequence of reactions within the mitochondrial matrix. Each reaction modifies the molecule slightly. Bonds are rearranged. Carbon atoms are released. Energy carriers are produced. But before following the sequence, it is important to slow the perspective. The cycle is not just a list of steps. It is a repeating structure that maintains the flow of energy through the cell. Each turn of the cycle produces intermediate molecules that carry high-energy electrons. These electrons are not used immediately. They are stored in carriers that will later transfer them to the inner membrane system. This separation of processes is important. The matrix prepares energy. The membrane converts it into usable form. Inside the cycle, carbon atoms are gradually released in the form of carbon dioxide. This release is not random. It occurs at specific steps in the cycle. Each release corresponds to a controlled oxidation reaction. Oxidation here means the loss of electrons. These electrons are not lost. They are transferred to carrier molecules such as NADH and FADH2. These carriers are essential intermediates. They act as temporary storage units for high-energy electrons. Once formed, these carriers leave the cycle and move toward the inner membrane. There, they will contribute to the electron transport chain described earlier. This separation of location is important. The cycle does not directly produce ATP in large amounts. Instead, it produces the raw materials for energy conversion. We can think of the cycle as a preparation system. It refines molecules and extracts usable energy in a stored form. The structure of the cycle itself is circular. After each complete turn, the starting molecule is regenerated. This allows the process to continue indefinitely as long as inputs are available. This circular nature is not symbolic. It reflects actual biochemical regeneration of intermediates. To understand why this is efficient, consider linear processes. In a linear process, starting material is consumed and must be replaced externally. In a circular process, internal components are reused. This reduces dependency on external regeneration of complex intermediates. Within the mitochondrial matrix, enzymes guide each step of the cycle. These enzymes are highly specific. Each enzyme catalyzes one reaction. This specificity ensures control. It prevents unwanted side reactions. It maintains the order of the cycle. As molecules pass through each step, they undergo subtle structural changes. Bonds are broken and reformed. Electrons are shifted between atoms. These changes are not dramatic in appearance. They are microscopic and gradual, but their cumulative effect is significant. Energy is extracted in small increments rather than large bursts. This controlled extraction prevents loss of energy as heat. It allows energy to be captured efficiently in carrier molecules. The cycle also produces molecules that can re-enter earlier metabolic pathways. This creates integration between different parts of cellular metabolism. Metabolism is not a set of isolated reactions. It is a network of interconnected cycles. The Krebs cycle sits at a central junction in this network. From a structural perspective, the cycle can be thought of as a rotating sequence of chemical states. Each state is defined by a specific molecular structure. Each transition is controlled by an enzyme. The matrix environment supports this process by maintaining suitable conditions. It provides enzymes, cofactors, and spatial organization. It also ensures that intermediates remain in proximity to the enzymes that process them. Diffusion within the matrix allows molecules to move between reaction sites. This movement is not directed like transport in larger systems. It is governed by concentration gradients and random motion. Yet, the system remains efficient due to spatial organization. As the cycle continues, energy carriers accumulate. NADH and FADH, two increase in number relative to their oxidized forms. This accumulation represents stored potential energy. But this energy is not yet usable for cellular work. It must be transferred to the inner membrane system where it can be converted into ATP. This division of labor is fundamental. The matrix prepares energy. The membrane converts it. The Krebs cycle also produces a small amount of ATP directly. However, this is not its primary output. Its main role is electron harvesting. Electrons are the key energy carriers in biological systems. Their movement through controlled pathways releases energy in usable forms. Within the cycle, electrons are carefully extracted from carbon-based molecules. These electrons are then transferred to carrier molecules. This transfer is facilitated by redox reactions. Redox reactions involve simultaneous oxidation and reduction. One molecule loses electrons, while another gains them. This exchange maintains balance. It ensures that electrons are not lost, but redistributed. The importance of this cycle becomes clearer when viewed as part of a larger system. It is not an isolated process. It is one stage in a continuous flow of energy conversion. Nutrients enter the cell. They are broken down into intermediate molecules. These molecules enter the mitochondrion. They are processed in the matrix. Energy carriers are produced. These carriers then move to the inner membrane. There, their stored energy is converted into a gradient. That gradient will later be used to produce ATP. This sequence is continuous. It does not pause. It adjusts based on cellular demand. If demand increases, cycle activity increases. If demand decreases, cycle activity slows. This regulation ensures efficiency. It prevents unnecessary energy production. The Krebs cycle is also a point of integration between different metabolic pathways. It receives inputs from carbohydrates, fats, and proteins. All these macronutrients eventually converge into common intermediates that enter the cycle. This convergence simplifies energy processing. Instead of separate pathways for each nutrient type, the cell uses a unified system. This design reflects efficiency in biological organization. As we observe this cycle more closely, it becomes clear that it is not merely a chemical sequence. It is a structured system of transformation and energy capture. Its circular nature ensures continuity. Its enzymatic control ensures precision. Its integration ensures efficiency. Within the mitochondrion, this cycle is constantly active, adjusting its speed and output based on cellular needs. It forms the foundation upon which membrane-based energy conversion can occur. Without it, the electron transport system would lack input. Without it, ATP production would slow significantly. Thus, the matrix cycle and the membrane system are inseparable parts of a larger energy network. As we transition forward, attention shifts from the matrix, where energy is prepared, to the inner membrane, where energy is transformed into gradients and stored in electrochemical form. The next section moves into the membrane-based processes, where electrons travel through protein complexes and energy is converted into a stored gradient across the mitochondrial inner membrane. At the inner mitochondrial membrane, energy becomes organized in a different form. It is no longer being extracted from carbon-based molecules. Instead, it is being transferred through a sequence of proteins embedded within a biological membrane. This sequence is not random. It is structured, directional, and tightly controlled. This system is known as the electron transport chain. To understand it clearly, we slow the perspective again. We are no longer in the matrix where molecules are broken down. We are now at the boundary where energy is converted into stored potential. The inner mitochondrial membrane is the stage for this process. It is a dense, folded surface containing multiple protein complexes. These complexes are arranged in sequence. Each one receives electrons and passes them forward. Electrons are not visible in isolation, but they represent energy at a fundamental level. Their movement through the chain is carefully managed. No step is uncontrolled. Electrons enter the chain from carrier molecules produced in the matrix, such as NADH and FADH2. These molecules release electrons at specific entry points. Once released, electrons move through a series of protein complexes embedded in the membrane. These complexes are often labeled as complex 1, 2, 3, and IV. Each complex has a specific role in transferring electrons and managing energy release. As electrons move from one complex to another, they lose energy gradually. This energy is not lost. It is captured and used to move protons across the membrane. Protons are hydrogen ions, positively charged particles. Their movement creates a difference in charge and concentration between two compartments of the mitochondrion. This difference is essential. It is the foundation of energy storage in the mitochondrion. To understand the system more clearly, we can think of the inner membrane as a controlled barrier between two environments. On one side is the matrix. On the other side is the intermembrane space. As electrons move through the chain, protons are actively transported from the matrix into the intermembrane space. This creates a higher concentration of protons outside the matrix. This concentration difference is not static. It is maintained by continuous electron flow. The result is an electrochemical gradient. This gradient represents stored potential energy. It is important to emphasize that this energy is not yet in the form of ATP. It is stored as a difference in distribution of particles across a membrane. This type of energy storage is efficient because it does not require large molecular changes. It relies instead on spatial separation and charge imbalance. The inner membrane is essential for maintaining this gradient. It is selectively permeable, meaning it restricts free movement of protons. Without this restriction, the gradient would collapse. The electron transport chain is therefore tightly linked to membrane structure. Without a controlled barrier, energy cannot be stored effectively. Within the chain, electrons pass through a series of redox reactions. Each transfer involves a donor molecule and an acceptor molecule. The donor loses electrons. The acceptor gains electrons. This exchange is carefully ordered so that energy is released in small increments. If energy were released all at once, it would be lost as heat. Instead, gradual transfer allows energy to be captured and used for proton movement. One of the key features of this system is coupling. Electron movement is coupled to proton pumping. This means that one process directly drives another. This coupling ensures efficiency. It prevents energy from being wasted. As electrons move through complex 1 and complex 3, energy is used to pump protons across the membrane. Complex 2 also participates in electron transfer, though it does not contribute directly to proton pumping in the same way. Complex IV completes the chain by transferring electrons to oxygen. Oxygen acts as the final electron acceptor. This step is essential. Without oxygen, electrons would not flow efficiently through the chain. When oxygen accepts electrons, it combines with protons to form water. This is a final stable product of the process. The formation of water marks the end of electron flow. It ensures that electrons do not accumulate in the system. At the same time, proton pumping continues to build the gradient. This gradient is stored across the inner membrane as potential energy. It is useful to think of this gradient as a form of pressure. Not physical pressure in the mechanical sense, but chemical and electrical tension between two regions. The matrix has a lower concentration of protons. The intermembrane space has a higher concentration. This difference creates a tendency for protons to move back toward equilibrium. However, the inner membrane prevents free diffusion. It forces protons to pass through specific channels. This restriction is what allows energy to be captured later. Without controlled movement, the gradient would dissipate without producing usable work. The electron transport chain does not directly produce ATP. Instead, it generates the conditions necessary for ATP synthesis. This distinction is important. Energy production in mitochondria is a multi-step process. Each stage contributes differently. The matrix prepares electrons. The electron transport chain moves electrons and builds a gradient. The final step converts that gradient into ATP. Within the membrane, the organization of protein complexes is not random. They are embedded in a way that allows efficient electron flow. Electron carriers move laterally within the membrane or within small diffusion distances. This ensures continuity of transfer. The membrane environment also provides stability for these complexes. Lipids surrounding the proteins help maintain structure and function. The interaction between proteins and membrane lipids is essential. It allows the system to remain flexible while maintaining integrity. As electrons move through the chain, energy is released in controlled steps. Each step contributes incrementally to proton movement. This incremental release is a key feature of biological energy systems. It allows fine control rather than abrupt changes. The accumulation of protons in the intermembrane space represents stored energy in a physical form. It is not stored as a molecule but as a gradient. This gradient is maintained as long as electron flow continues. When electron flow stops, the gradient gradually dissipates. This dependence on continuous flow highlights the dynamic nature of mitochondrial energy systems. Nothing is permanently static. Everything is maintained through ongoing activity. The electron transport chain is therefore not just a sequence of reactions. It is a continuous system of energy conversion and storage. It links the breakdown of nutrients in the matrix to the production of ATP in the final stage. Without this system, the energy extracted from nutrients would not be efficiently captured. It would be dispersed as heat or lost in uncontrolled reactions. Instead, mitochondria convert that energy into a structured gradient that can be used on demand. At this point, we can see mitochondria as systems that transform molecular energy into electrochemical storage. But this stored energy is still not in usable form for most cellular processes. It must pass through one final transformation. That transformation occurs through a membrane-bound enzyme complex that converts the proton gradient into ATP. Before moving to that step, it is important to recognize the elegance of this system. Energy is not created or destroyed. It is transferred through structured pathways, captured in gradients, and then converted into usable chemical bonds. This layered organization allows life to maintain order in a constantly changing environment. As we move forward, attention shifts from electron flow to proton flow. and from stored gradients to mechanical-like enzymatic conversion. The next section moves into the final stage of mitochondrial energy production, where the proton gradient is converted into ATP through a rotating molecular mechanism embedded in the inner membrane. At this final stage of mitochondrial energy conversion, the system shifts from storing potential energy to releasing it in a controlled, usable form. The gradient that was built across the inner membrane is now used directly. This is where stored difference becomes movement, and movement becomes chemical energy. To understand this clearly, we return once again to the inner mitochondrial membrane. It is here that the proton gradient exists, with a higher concentration of protons in the intermembrane space and a lower concentration in the matrix. This difference represents stored energy. But energy in this form is not directly usable by most cellular processes. It must be converted into ATP, a molecule that can be transported and used throughout the cell. The conversion process is performed by a specialized enzyme complex embedded in the inner membrane. This complex is called ATP synthase. ATP synthase is not a static structure. It is dynamic. It changes shape during its function. It responds directly to the movement of protons. To slow the understanding, we can think of ATP synthase as a molecular machine that uses flow to generate chemical bonds. But even this analogy is limited. It is more accurate to describe it as a coupled system where chemical gradients drive conformational changes. The structure of ATP synthase spans the inner mitochondrial membrane. One part of it sits within the membrane, forming a channel. Another part extends into the matrix, where ATP production occurs. The membrane-embedded portion allows protons to pass through a controlled pathway. This movement is not free diffusion. It is guided through a specific channel. As protons move through this channel, they cause physical rotation in part of the ATP synthase complex. This rotation is not symbolic. It is a mechanical movement at the molecular scale. This rotational motion is the key to ATP production. It links the flow of protons to the synthesis of a high-energy molecule. To understand why rotation is involved, it helps to slow down and consider the principle behind the system. The proton gradient represents stored potential energy. When protons move back toward equilibrium, they release this energy. ATP synthase captures this movement. Instead of allowing protons to simply diffuse back into the matrix, it channels their movement through a structure that converts flow into motion. This motion is then used to drive chemical reactions in the catalytic portion of the enzyme. Inside ATP synthase, there are multiple subunits that change shape depending on rotational position. These shape changes allow the enzyme to bind ADP and inorganic phosphate and then combine them into ATP. ADP, or adenosine diphosphate, is a lower energy molecule. It has 2 phosphate groups. ATP has 3 phosphate groups. The addition of the 3rd phosphate group requires energy input. That energy comes from the proton gradient. As protons move through ATP synthase, they cause conformational changes that make the binding and formation of ATP energetically favorable. This process is not a simple addition reaction. It is a coordinated cycle of binding, change, and release. ATP synthase operates in repeated cycles. Each cycle produces multiple ATP molecules. The exact number depends on proton flow and gradient strength. The important principle is that energy is not directly transferred into ATP in a single step. Instead, it is mediated through mechanical-like motion generated by chemical gradients. This coupling of gradient to motion is one of the most efficient energy conversion systems in biology. To visualize the system slowly, imagine protons accumulating outside the matrix. They are held back by the inner membrane. When they pass through ATP synthase, they are allowed to return to equilibrium. But this return is not free. It is harnessed. The energy of their movement is captured and redirected into enzymatic activity. This redirection is what allows life to convert gradients into usable energy. The inner membrane, therefore, serves two roles at once. It maintains the gradient and provides the pathway for controlled release. Without this dual function, energy storage and energy use would not be connected. ATP produced by ATP synthase is then released into the matrix. From there, it is transported to other parts of the cell. ATP is the primary energy currency of the cell. It is used in many processes, including muscle contraction, protein synthesis, and transport across membranes. Every time ATP is used, it is converted back into ADP. This creates a continuous cycle of energy use and regeneration. Mitochondria are responsible for maintaining this cycle. They replenish ATP as it is consumed. The efficiency of ATP synthase is remarkable. It operates with a high degree of coupling between proton movement and ATP production. This means that most of the energy from the gradient is converted into chemical energy rather than being lost as heat. However, no biological system is perfectly efficient. Some energy is always lost during conversion. This is a fundamental property of physical systems. Even so, the system remains highly optimized for biological function. ATP synthase is often described as one of the most efficient molecular machines in biology. Its ability to convert a gradient into chemical energy is central to cellular life. The importance of this final step cannot be overstated. Without ATP synthesis, the energy extracted in earlier stages would remain inaccessible. The matrix cycle produces electrons. The electron transport chain creates a gradient. ATP synthase converts that gradient into usable energy. These three stages form a continuous system. Each depends on the previous one. If electron production stops, the chain stops. If the chain stops, the gradient collapses. If the gradient collapses, ATP production stops. This dependency highlights the integrated nature of mitochondrial function. Inside the cell, ATP production is not stored in large reserves. It is continuously generated and consumed. This continuous turnover allows the cell to respond quickly to changes in energy demand. When demand increases, ATP synthase activity increases indirectly through stronger proton gradients. When demand decreases, electron flow slows and ATP production adjusts accordingly. This responsiveness ensures balance. The coupling between proton flow and ATP production is also an example of energy transduction. Energy is transferred from one form to another without being stored in intermediate, unstable forms. This minimizes waste and increases efficiency. At the molecular level, ATP synthase demonstrates that biological systems can convert physical gradients into chemical bonds through structured mechanical-like processes. This is not mechanical in the classical sense, but it behaves in a way that resembles mechanical systems. Motion drives change. Change drives synthesis. The result is ATP, a stable molecule that can be transported and used throughout the cell. Once ATP is produced, it leaves the mitochondria and enters the cytoplasm. There, it participates in countless cellular reactions. Each ATP molecule represents a small packet of usable energy. When broken down, it releases energy that powers cellular work. This work includes movement, synthesis, transport, and signaling. In this way, mitochondria serve as central hubs in the flow of biological energy. They do not store large amounts of ATP directly. Instead, they maintain the system that continuously produces it. As we step back from the molecular detail, we can see a complete system. Nutrients become electrons. Electrons create a gradient, and the gradient produces ATP. Each step is dependent on structure, organization, and controlled movement. The mitochondrion is therefore not a single function unit. It is a layered conversion system that transforms chemical energy into biological activity. As we continue forward, attention shifts beyond energy production alone. Mitochondria also influence signaling, regulation, and cellular decisions that extend beyond ATP synthesis. The next section moves beyond energy production into broader mitochondrial roles in cellular regulation, including signaling, stress response, and controlled cell life cycle processes. Energy production inside mitochondria can appear complete once ATP has been formed. Nutrients have been broken down. Electrons have moved through membrane complexes. A proton gradient has formed and been converted into ATP. But the story does not end there. Because no energy system operates in isolation. It must be maintained, adjusted, protected, and integrated into a changing cellular environment. This is where another layer of mitochondrial function appears. Not the creation of energy, but the regulation of how energy systems remain stable. To understand this, we begin with a simple principle. Any system that produces energy also produces stress. This is true in physical systems. It is also true in biological systems. Inside mitochondria, the movement of electrons is highly controlled. But no transfer system is perfectly closed. A small fraction of electrons can sometimes react prematurely with oxygen. When this happens, reactive oxygen species may form. These are chemically reactive molecules derived from oxygen. At low levels, they are not simply harmful byproducts. They can also function as signaling molecules. This is an important distinction. Biology often uses molecules in multiple roles. A molecule can be both potentially damaging and functionally useful, depending on amount and context. In controlled amounts, reactive oxygen species can participate in signaling pathways that help cells adapt to conditions. But if levels rise excessively, they can contribute to oxidative stress. Oxidative stress refers to imbalance between reactive molecules and protective systems that neutralize them. Mitochondria contain protective mechanisms to help maintain this balance. Enzymatic systems help convert reactive species into less harmful forms. This is part of mitochondrial maintenance. Energy production must be paired with protective regulation. Without this regulation, efficiency would decline over time. Molecular components could accumulate damage. So mitochondria do more than generate ATP. They help preserve the conditions under which ATP generation can continue reliably. This introduces a broader idea. Mitochondria are not only engines of production. They are systems of maintenance. Maintenance operates at multiple levels. It includes managing reactive molecules. It includes monitoring membrane integrity. It includes replacing damaged components. Consider the inner membrane. Its structure is essential for maintaining proton gradients. If membrane integrity weakens, gradient efficiency falls. ATP production can become less effective. Cells therefore maintain and repair mitochondrial components continuously. Proteins involved in electron transport can be replaced. Lipid components of membranes can be renewed. Damaged molecular parts can be removed. This constant renewal is easy to overlook. Yet it is essential. Biological systems endure not because they resist wear completely, but because they repair continuously. Mitochondria follow this principle. There is also quality control at the level of entire mitochondria. Individual mitochondria can change shape, merge together, or divide apart. These processes are called fusion and fission. Fusion allows mitochondria to combine contents. This can help distribute components and support functional balance. Fission allows mitochondria to divide. This contributes to distribution, renewal, and quality control. These are not random shape changes. They are regulated processes. Through fusion and fission, mitochondrial populations remain adaptable. They can respond to stress, energy demand, and damage. This dynamic behavior reveals something important. Mitochondria are not static bean-shaped structures frozen inside textbook diagrams. They are dynamic participants in cellular life. They move. They reshape. They renew. And sometimes, when damage is too extensive, cells can selectively remove dysfunctional mitochondria. This process is often called mitophagy. It is a form of targeted quality control. Rather than allowing damaged mitochondria to persist and disrupt cellular balance, the cell can identify and recycle them. Again, this is not failure. It is maintenance. Removal of damaged components is often a sign of healthy regulation, not breakdown. Through these processes, mitochondrial populations are constantly being monitored and renewed. This makes energy production a living system of upkeep, not merely a sequence of reactions. Another layer of regulation involves uncoupling. Earlier, proton gradients were described as tightly linked to ATP synthesis. In many contexts, that coupling is strong and efficient. But in some cases, protons can return across the membrane without driving ATP synthase directly. This can release energy as heat. This process is known as mitochondrial uncoupling. It may sound inefficient, but in some contexts it has useful roles, including thermoregulation and modulation of reactive oxygen production. This reminds us that biological efficiency is not always about maximizing a single output. Sometimes flexibility and protection matter more than raw efficiency. Mitochondria operate within that broader logic. They optimize not merely for ATP yield, but for stability of the whole system. This idea is subtle but important. Biological systems often prioritize resilience over theoretical maximum efficiency. A slightly less efficient but more stable system may be favored over a fragile, highly optimized one. Mitochondrial regulation reflects this principle repeatedly. Another aspect of maintenance involves communication with the rest of the cell. Mitochondria do not regulate themselves in isolation. Signals pass between mitochondria and the nucleus, coordinating responses to stress and energy demand. This communication helps adjust protein production, repair pathways, and metabolic priorities. Again, energy production is embedded within larger regulatory networks. This becomes especially important under changing conditions. During increased energy demand, mitochondria may increase activity. During stress, protective responses may strengthen. The system shifts continuously. This shifting is not instability. It is adaptive regulation. We can now begin to see mitochondria in a broader way, not merely as power producers, but as self-maintaining energetic networks. Production and protection are inseparable. Energy generation creates the need for regulation. Regulation preserves the capacity for energy generation. The relationship is circular, much like the metabolic cycles themselves. Even at the level of population behavior inside a cell, mitochondria can form networks. In some cells, they appear as interconnected reticular structures rather than isolated units. This organization may support communication and distribution of resources. Again, structure and function remain linked. Dynamic networks allow flexibility. Flexibility supports resilience. Resilience supports continuity of life processes. At this point, the concept of mitochondria has become wider than the familiar phrase powerhouse of the cell. That phrase captures something true but incomplete. Mitochondria are not only powerhouses. They are managers of energetic stability. They are guardians of internal balance. And this prepares the ground for an even broader role. Because once an organelle can sense energy state, manage stress, regulate damage, and communicate with the cell, it begins participating in decisions about the life of the cell itself. That larger regulatory role extends beyond maintenance. into signaling and control of cellular fate. The next section moves from mitochondrial maintenance and quality control into how mitochondria participate in signaling networks, stress responses, and broader regulation of cellular survival. Mitochondria are often introduced through their role in energy production. But energy is only one aspect of their function. Over time, research has shown that mitochondria are also deeply involved in cellular regulation. They participate in signaling, stress response, and the coordination of cell survival and cell death. To understand this broader role, it is useful to step away from the idea of mitochondria as isolated energy units. Instead, we view them as integrated sensors and regulators within the cell. They do not only produce ATP, they continuously respond to the internal condition of the cell. This responsiveness is based on chemical signals. Mitochondria detect changes in energy demand, nutrient availability, oxygen levels, and cellular stress. These signals influence mitochondrial behavior at multiple levels. One of the most important regulatory roles of mitochondria is calcium handling. Calcium ions act as signaling molecules inside cells. Small changes in calcium concentration can trigger large changes in cellular behavior. Mitochondria can absorb and release calcium ions. This allows them to help shape calcium signals within the cell. When calcium levels rise in the cytoplasm, mitochondria can take up some of these ions. This helps prevent excessive signaling and contributes to balance. Later, mitochondria can release calcium back into the cytoplasm when needed. This controlled exchange contributes to timing and regulation of cellular processes. In this way, mitochondria are not passive energy producers. They are active participants in signaling networks. Another important aspect of mitochondrial regulation is their response to metabolic state. Metabolism refers to the collection of chemical reactions that sustain life. Mitochondria respond to whether the cell is in a high energy or low energy state. When energy is abundant, mitochondrial activity can shift toward storage and maintenance functions. When energy is low, mitochondrial activity shifts toward increased ATP production. This flexibility is achieved through feedback mechanisms. Molecules such as ATP, ADP, and NADH serve as indicators of energy status. Their concentrations influence mitochondrial processes. For example, high levels of ATP can signal that energy demand is currently low. This can reduce the rate of electron transport and ATP synthesis. Conversely, high levels of ADP indicate that ATP is being consumed. This increases mitochondrial activity to restore balance. This feedback system ensures that energy production is matched to demand. It prevents unnecessary waste of resources. Mitochondria also respond to oxygen availability. Oxygen is the final electron acceptor in the electron transport chain. Without oxygen, electron flow slows or stops. When oxygen levels are low, mitochondria adjust their activity. They may reduce electron transport and shift metabolic pathways. This adaptation is important for cell survival under varying environmental conditions. Beyond energy and signaling, Mitochondria are also involved in managing cellular stress. Stress in this context refers to conditions that disrupt normal cellular function, such as oxidative imbalance, nutrient limitation, or damage to molecules. One type of stress related to mitochondria is oxidative stress. During electron transport, small amounts of reactive oxygen species can be produced. These molecules can damage proteins, lipids, and DNA if not controlled. Mitochondria contain systems to manage and neutralize these reactive molecules. This helps maintain cellular stability. However, if oxidative stress becomes too high, it can trigger broader cellular responses. One of the most significant regulatory roles of mitochondria is their involvement in programmed cell death, also known as apoptosis. Apoptosis is not a chaotic process. It is a controlled and organized way for cells to be removed when they are no longer functional or when they pose a risk to the organism. Mitochondria play a central role in initiating this process. When a cell experiences severe damage or stress, mitochondria can release specific signaling molecules. These molecules trigger a cascade of reactions inside the cell. This cascade leads to systematic breakdown of cellular components. This process ensures that damaged cells are removed in a controlled manner without causing harm to surrounding tissue. It is important to understand that apoptosis is not a failure of mitochondria. It is a regulated function. It contributes to overall organism health by maintaining cellular quality control. In this way, mitochondria are involved in decisions at the cellular level. Not decisions in a conscious sense, but biochemical decisions based on internal conditions. They integrate signals related to energy, damage, and environmental stress. Based on these signals, they contribute to determining whether a cell continues functioning or initiates self-removal. This regulatory role highlights the complexity of mitochondrial function. They are not single-purpose organelles. They are multifunctional systems embedded in cellular regulation. Mitochondria also interact with the nucleus in signaling pathways. This interaction is bidirectional. The nucleus sends instructions that influence mitochondrial protein production. Mitochondria send signals that influence nuclear gene expression. This communication ensures coordination between energy production and cellular needs. For example, when mitochondria experience stress, they can signal the nucleus to activate protective genes. These genes help restore balance or adapt to new conditions. This feedback loop creates a dynamic relationship between energy systems and genetic regulation. Mitochondria also influence metabolic flexibility. Cells can use different types of fuel depending on availability. Mitochondria help determine which fuel sources are used efficiently. This flexibility is important for survival in changing environments. In muscle cells, for example, mitochondria help adjust energy production during rest and activity. During activity, demand increases rapidly. Mitochondria respond by increasing ATP production. During rest, energy demand decreases. Mitochondria reduce activity accordingly. This dynamic adjustment ensures that energy supply matches physiological demand. Mitochondria also participate in shaping cellular lifespan. Cells do not live indefinitely. Their lifespan is influenced by accumulated damage, energy efficiency, and regulatory signals. By influencing apoptosis and stress responses, mitochondria contribute to determining when a cell should be replaced. This replacement is part of normal biological maintenance. It allows tissues to remain functional over time. In addition, mitochondria are involved in temperature regulation in certain tissues. Some mitochondrial processes can release energy as heat instead of ATP. This is important in maintaining body temperature in certain physiological conditions. This shows again that mitochondria are not limited to one output. They can adjust energy flow based on biological context. Across all these roles, energy production, calcium signaling, stress response, apoptosis, and metabolic regulation, mitochondria function as integrated hubs. They sit at the intersection of chemistry and regulation. They interpret internal conditions and adjust cellular behavior accordingly. This interpretation is not conscious. It is biochemical. It arises from networks of molecular interactions that respond predictably to changing conditions. As we step back from the details, a broader picture emerges. Mitochondria are not just energy producers. They are central integrators of cellular state. They connect metabolism, signaling, and survival into a single coordinated system. This integration allows cells to remain stable in a changing environment. As we move forward, attention shifts toward the idea that mitochondria do not operate in isolation even within a single cell type. Their behavior varies depending on tissue, function, and energy demand across the body as a whole. The next section moves outward from single-cell regulation into how mitochondrial behavior differs across tissues, and how this variability supports the functioning of the entire human body. Across the human body, mitochondria are not identical in behavior or density. They follow a general principle, but their expression adapts to the needs of each tissue. This variation is not random. It reflects functional demand. Different cells require different amounts of energy. Some cells perform continuous mechanical work. Some process information. Some maintain long-term structural stability. Because of this, mitochondrial distribution and activity vary across the body. In muscle tissue, mitochondria are abundant. Muscle cells require constant ATP for contraction and relaxation cycles. This demand is continuous, even at rest. In neurons, mitochondria are also highly active. Neurons require energy to maintain ion gradients and transmit electrical signals. These processes occur rapidly and repeatedly. In contrast, some connective tissues have lower energy demands. Mitochondria are still present, but less densely concentrated. This variation demonstrates a key principle of biological organization. Structure follows function. Mitochondria are not uniformly distributed because energy demand is not uniform. To understand this more deeply, we can view the body as a network of energy requirements. Each tissue represents a node in this network. Each node has a different level of demand. Mitochondria adapt to these demands by adjusting number, activity, and efficiency. This adaptability is essential for organism-level stability. Without it, some tissues would experience energy deficits, while others would have excess capacity. At the cellular level, this adaptation is achieved through regulation of mitochondrial biogenesis and degradation. Cells can increase mitochondrial numbers when energy demand increases. They can also remove damaged mitochondria when necessary. This dynamic turnover maintains mitochondrial quality and efficiency. The process of creating new mitochondria is controlled by genetic and metabolic signals. It involves coordination between nuclear DNA and existing mitochondria. Similarly, damaged mitochondria are removed through a process that ensures cellular stability. This prevents accumulation of inefficient or harmful components. At a larger scale, mitochondrial variation across tissues supports specialization. In the heart, for example, mitochondria are extremely dense. The heart requires continuous contraction throughout life without rest. This demands a constant and reliable ATP supply. In skeletal muscle, mitochondrial density is also high, but varies depending on muscle type. Endurance muscles contain more mitochondria than muscles designed for short bursts of activity. This reflects different energy strategies. Endurance relies on sustained ATP production. Short bursts rely more on rapid energy systems with different metabolic pathways. In the brain, mitochondrial function is tightly regulated. Neurons are highly energy dependent due to continuous signaling activity. Even small disruptions in mitochondrial function can affect neural performance. This sensitivity highlights the importance of mitochondrial regulation in cognitive processes. Across all tissues, mitochondria serve the same fundamental role. They convert nutrients into usable energy. But the scale and intensity of this role differ. This difference is not only structural, but also functional. It reflects how each tissue interacts with energy over time. To understand this system at a deeper level, it is useful to consider energy demand as a continuous variable rather than a fixed state. At any moment, tissues experience changing levels of activity. These changes require rapid adjustment in ATP supply. Mitochondria respond to these changes through metabolic flexibility. They adjust electron transport rates, proton gradient strength, and ATP synthesis accordingly. This responsiveness ensures that energy production remains aligned with real-time demand. If we step back further, we can see that mitochondria are part of a larger systemic balance. The human body maintains homeostasis, a stable internal environment despite external changes. Mitochondria contribute to this stability by regulating energy availability. Energy is not stored in a single location. It is distributed across cells and tissues. Mitochondria ensure that this distribution remains functional. In tissues with high demand, mitochondria are more active. In tissues with lower demand, they reduce activity. This balance is maintained continuously. Another important aspect of mitochondrial variation is metabolic specialization. Different tissues rely on different fuel sources depending on their function. Some tissues prefer glucose. Others rely more on fatty acids. Mitochondria adapt to process these fuels efficiently. This adaptability is achieved through enzyme regulation within the matrix and membrane systems. The ability to switch between fuel types is essential for survival during changing nutritional conditions. For example, during fasting, the body shifts toward increased fat utilization. Mitochondria in certain tissues adjust accordingly. This metabolic flexibility allows the organism to maintain energy balance even when external inputs change. Mitochondrial differences across tissues also reflect developmental programming. During development, cells differentiate into specialized types. This differentiation includes changes in mitochondrial structure and function. As cells specialize, their energy systems become tailored to their roles. This specialization is maintained throughout life, but remains adaptable. Mitochondria also play a role in aging processes. Over time, mitochondrial efficiency can decline due to accumulated molecular damage. This affects energy production capacity. Cells and tissues with high energy demand are particularly sensitive to these changes. However, cells also have mechanisms to repair or replace damaged mitochondria. This helps maintain function over time. The balance between damage and repair is a key factor in cellular aging. At the organism level, mitochondrial function contributes to overall vitality. Efficient energy production supports movement, cognition, repair, and adaptation. When mitochondrial function is well regulated, systems remain stable. When it is disrupted, multiple systems can be affected simultaneously. This is because mitochondria are present in nearly all cell types. Their influence is therefore widespread. Despite this influence, mitochondria do not operate independently of cellular context. Their behavior is always shaped by signals from the surrounding environment. This ensures coordination between local energy production and systemic needs. If we view the body as a whole system, mitochondria act as distributed energy processors. They are embedded within every major functional unit. This distribution allows energy production to occur close to where it is needed. This reduces delays and increases efficiency. It also allows for fine-tuned regulation at the local level. Each tissue can adjust mitochondrial behavior independently while still contributing to overall organism balance. This multi-level regulation is one of the defining features of biological systems. Mitochondria are therefore not just cellular components. They are part of a hierarchical energy system that spans from molecules to organs. Their variation across tissues reflects the diversity of biological function. As we move toward the final perspective, we begin to see mitochondria not only as energy converters or regulators, but as continuous presences within every moment of biological activity. They operate silently, constantly, and adaptively across the entire body. The next section moves toward synthesis, stepping back from individual systems to observe mitochondria as a continuous presence underlying all cellular life, connecting energy, regulation, and biological continuity into a single, unified perspective. When we step back from individual tissues and individual processes, a different picture begins to form. Instead of seeing mitochondria as separate components performing isolated tasks, we begin to see them as a continuous presence distributed across all living cells. This presence is not centralized. It is widespread, embedded, and persistent. Every active cell contains mitochondria. Every tissue with metabolic demand depends on them. Every moment of biological activity is supported by their function. This universality creates a unifying layer across all biological systems. Despite differences in function, structure, and specialization, all cells share a dependency on mitochondrial energy conversion. To understand this more clearly, it helps to slow down the perspective again. We are no longer looking at individual cycles or mechanisms. We are observing continuity. Continuity in biology means that processes do not begin and end in isolation. They flow into one another. They overlap in time and scale. Mitochondria are part of this continuous flow. They do not operate in short bursts of independent activity. They operate in sustained, ongoing cycles of energy conversion. From a molecular perspective, this continuity is maintained through constant turnover of substrates and products. Nutrients are continuously processed. Electrons are continuously transferred. Gradients are continuously formed and dissipated. ATP is continuously produced and consumed. This repetition is not redundancy. It is stability. Stability in living systems is not achieved through stillness. It is achieved through regulated motion. Mitochondria embody this principle. They maintain energy stability through continuous transformation. If we expand this view further, we can see that mitochondrial activity is always present at some level in all aerobic life. Even during rest, even during sleep, even during minimal activity, mitochondrial processes continue. This persistence reflects the fundamental requirement of energy in biological systems. Life is not a static state. It is a maintained condition that requires constant energy input. Mitochondria provide that input in a structured form. At the same time, mitochondria are not uniform across all cells. Their behavior is shaped by context. This creates variation within continuity. Continuity ensures that energy is always available. Variation ensures that energy is appropriately distributed. Together, these principles form a balanced system. To understand mitochondria at this level, it is useful to think in terms of layered timescales. Some mitochondrial processes occur in milliseconds. Others occur over seconds, minutes, or longer periods. Electron transport is extremely rapid. ATP synthesis follows closely. Metabolic regulation occurs over slightly longer time frames. Structural adaptation of mitochondria occurs over even longer periods. This layering of time creates a multi-scale system. At every moment, mitochondria are operating across different temporal levels simultaneously. This is part of what makes biological systems complex. They are not governed by a single timescale. They are governed by interacting timescales. Within this framework, mitochondria serve as a bridge between fast molecular events and slower cellular regulation. For example, electron transfer occurs almost instantly at the molecular level. But the resulting ATP production influences processes that occur over much longer durations, such as muscle contraction, protein synthesis, or neural signaling. This connection between fast and slow processes is essential. Without it, biological systems would lose coordination. Mitochondria therefore function as translators between different levels of biological time. They convert rapid chemical reactions into sustained biochemical availability. When we observe mitochondria across the entire body, another pattern emerges. They are not evenly distributed in a static sense, but they are consistently present in proportion to demand. This proportionality is dynamic. It changes over time, depending on physiological conditions. For example, during increased physical activity, certain tissues increase mitochondrial activity. During recovery, this activity adjusts again. This responsiveness ensures that the body remains balanced under changing conditions. Mitochondria are therefore not fixed energy units. They are adaptable systems embedded within a responsive network. This network spans all cells, all tissues, and all energy-dependent processes. At a deeper level, mitochondria also contribute to maintaining internal order within cells. They influence the balance between synthesis and breakdown of molecules. They regulate whether resources are used immediately or stored for later use. This regulation is essential for preventing instability. If energy production were uncontrolled, cells would experience imbalance. If energy production were insufficient, cells would fail to function. Mitochondria maintain this balance through continuous adjustment. Another aspect of mitochondrial continuity is their involvement in cellular adaptation. Cells must respond to environmental changes. These changes may include nutrient availability, oxygen levels, temperature, or mechanical stress. Mitochondria help cells adjust to these conditions by modulating energy production and signaling pathways. This adaptability allows cells to survive in changing environments without losing function. At the organism level, this adaptability becomes even more significant. It allows the body to maintain homeostasis across a wide range of conditions. Homeostasis is not a static state. It is a dynamic balance maintained through continuous adjustment. Mitochondria are central to this adjustment process. They continuously respond to internal signals and external conditions. This responsiveness creates a feedback loop between energy production and physiological demand. In this loop, mitochondria adjust output based on signals, and the resulting energy availability influences future signals. This recursive interaction is a defining feature of biological regulation. When viewed across time and scale, mitochondria represent a constant background process. They are always active, always responsive, and always integrated into cellular function. This does not mean they are unchanging. It means their function is continuous rather than episodic. Even when the body appears still, mitochondrial processes continue at a baseline level. This baseline is essential for maintaining life. Without it, cellular systems would quickly lose stability. As we consider this continuous presence, it becomes clear that mitochondria are not simply components within cells. They are ongoing processes distributed across all cellular environments. They represent a form of biological continuity that links molecular reactions to organism-level function. This continuity is not visible at a single moment. It becomes apparent only when observed across time and scale. From this perspective, mitochondria can be understood as the underlying energy rhythm of life. Not a single rhythm in isolation, but a distributed pattern repeated across trillions of cells. Each cell contributes to this pattern. Each mitochondrion participates in maintaining it. This creates a system that is both localized and global within the body. Localized in that each cell manages its own energy production. Global in that all cells together form a unified metabolic system. Mitochondria sit at the center of this duality. As we move toward the final perspective, we begin to see mitochondria not just as energy systems or regulators, but as continuous threads connecting every biological process. They are present in every moment of cellular activity. They support every transition from molecular energy to biological function. The next section moves into a final synthesis, bringing together energy production, regulation, and continuity into a unified view of mitochondria as the persistent energetic foundation underlying all living cellular systems. When all layers are brought together, mitochondria can be understood as a single integrated system expressed across many scales. From molecules to cells, from tissues to the whole organism, they form a continuous energetic foundation. Not as a visible structure in motion, but as a persistent process embedded in life itself. This final perspective is not about adding new mechanisms. It is about unifying what has already been observed. Energy production, electron transfer, proton gradients, ATP synthesis, signaling, regulation, and tissue variation are not separate stories. They are expressions of the same underlying system. To understand this clearly, we begin again from the simplest point. A living cell requires energy. This energy cannot be stored indefinitely in unstable forms. It must be generated continuously and delivered in usable packets. ATP serves this role. It is the immediate energy currency of the cell. But ATP itself is not the source. It is the output of a deeper system. That deeper system is mitochondrial function. Inside mitochondria, nutrients are broken down into electrons. These electrons are transferred through structured protein complexes. Their movement builds a proton gradient across the inner membrane. This gradient stores energy in a spatial form. It is not chemical bond energy alone. It is separation of charge and concentration. Then, ATP synthase converts this stored gradient into chemical energy. It does this through controlled molecular motion and enzymatic transformation. This sequence is linear in description but continuous in reality. It loops without a defined beginning or end. Electrons flow as long as nutrients are available. Gradients form as long as electrons flow. ATP is produced as long as gradients exist. This is not a chain of isolated events. It is a coupled system of interdependent flows. At the same time, mitochondria do not function solely as energy converters. They also act as regulators of cellular state. They sense ATP levels, oxygen availability, calcium concentration, and metabolic stress. They respond by adjusting their internal processes. This response is not symbolic. It is biochemical adjustment through enzyme activity, gene signaling, and membrane dynamics. In this way, mitochondria function as feedback nodes within the cell. They do not only produce energy, they also interpret energy status. This interpretation influences cellular decisions such as growth, repair, adaptation, or programmed cell death. When viewed across multiple cells, this role becomes even more significant. Mitochondria contribute to coordination between tissues. They help ensure that energy production matches organism-wide demand. In high-demand tissues, mitochondrial density increases. In low-demand tissues, it decreases. This distribution is not fixed. It adjusts over time. This adjustment reflects a deeper principle of biological systems. Function is maintained through adaptability rather than rigidity. Mitochondria embody this principle at every level. At the molecular level, they adapt through enzyme regulation. At the cellular level, through signaling pathways. At the tissue level, through changes in density and activity. At the organism level, through systemic metabolic coordination. These layers are not separate. They are nested expressions of the same system. This nesting creates a hierarchy of energy control. Small-scale processes influence large-scale outcomes. Large-scale conditions influence small-scale behavior. Mitochondria sit within this hierarchy as both local processors and global contributors. They are local in that each mitochondrion responds to its immediate environment. They are global in that their collective activity determines organism-level energy availability. This dual nature is essential for life. Without local control, cells could not respond to immediate needs. Without global coordination, the organism could not maintain balance. Mitochondria bridge this gap continuously. Another key aspect of this unified system is time. Mitochondria operate across multiple temporal scales simultaneously. At the fastest scale, electrons move through protein complexes in fractions of a second. At intermediate scales, ATP is produced and consumed in cycles of activity. At longer scales, mitochondrial number and efficiency adjust through biogenesis and turnover. These time layers interact. Short-term changes influence long-term adaptation. Long-term conditions shape short-term responses. This temporal integration allows mitochondria to maintain stability in a changing environment. Stability in biology is not static balance. It is dynamic equilibrium maintained through continuous adjustment. Mitochondria are central to this equilibrium. They ensure that energy is always available, but never uncontrolled. They ensure that energy is always responsive, but never chaotic. When viewed from this perspective, Mitochondria are not just organelles. They are regulatory frameworks embedded within cells. They define how energy flows through biological systems. They shape how cells respond to internal and external conditions. They influence how tissues function together as a coordinated organism. This makes them foundational to life as it is currently understood in aerobic organisms. Every heartbeat, every neural impulse, every muscle contraction, every cellular repair process depends on mitochondrial output. Even processes that seem independent of energy production are ultimately supported by mitochondrial activity. This includes signaling pathways, structural maintenance, and molecular synthesis. At the most unified level, mitochondria can be seen as the interface between chemical energy and biological organization. They convert environmental resources into structured internal order. This conversion is not a one-time event. It is continuous. It is maintained moment by moment across the entire lifespan of a cell. This continuity is what allows life to persist in a state far from equilibrium. Without continuous energy input and regulation, biological systems would naturally drift toward disorder. Mitochondria prevent this drift by sustaining organized energy flow. They are therefore not just contributors to life. They are maintainers of its structure. As we bring this synthesis to a close, the image that remains is not of isolated mechanisms, but of a distributed, energetic network. A network that exists within every cell. A network that connects molecular reactions to organism-level function. A network that operates continuously without interruption. Mitochondria are the active nodes of this network. They do not merely support life. They sustain the energetic conditions that make life possible. And as long as these conditions are maintained, the system continues. The final transition now moves beyond description of structure and function toward a reflective synthesis of how continuous mitochondrial activity forms the invisible background rhythm underlying all living biological processes.